Hey guys, so this is the very last video in chapter two. Chapter two covers a ton of concepts. It's a ton of material, organic molecules, and all of that is involved with that organic chemistry, water, and now we're going to talk about macromolecules. So this is, if we were in Majors Bio in 1107, this is a few different chapters combined into one chapter here for 1010. So these are some major concepts that we just kind of touch the surface of, where if we were in Majors Bio, we would go into more detail on these things. But let's go through this very last section on macromolecules, biological molecules. Let's jump in. So to begin with these macromolecules, macro means big, right? So these are going to be really big molecules after we started about the teeny tiny guys and atoms and putting all these guys together. So when we talk about putting things together and breaking things apart, that's your metabolism. And there's two different reactions that happen in your metabolism. And your metabolism's going all day, every day, 24/7. You're going to have two pathways. You're going to have an anabolic where you're going to take small things and you make a bigger thing. Small things and you make a bigger thing. Two bond these things together, to take the small thing and this small thing and bond them together, that requires energy. So energy has to be added into this system to create your larger, to synthesize your larger molecule. Here's our larger molecule over here. So if we were talking about plants and photosynthesis, for example, if we took the CO2 molecules, which are small, and the water molecules, which are relatively small, and we add in the sun solar energy at the end of photosynthesis, we end up with glucose, which is a bigger molecule than our original smaller counterpart of CO2 and water. So anabolic is going to take small things and make big things. If you want to think about anabolic like anabolic steroids and how people use those to bulk up and create bigger muscles, whatever helps you remember that, anabolic is taking small things and synthesizing a bigger molecule. Reverse this, we're going to flip it. Catabolic, cats have claws. My two cats like to shred furniture. They like to shred furniture. They've shred two sofas so far. So they like to take my gigantic sofa and shred the fabric into teeny tiny little strips. So if you take a very large molecule, well, not necessarily very large, but if you take a large molecule and you break it into smaller things, you take a big thing and you break it into smaller things. Here's our big guy, and here's our smaller molecule. When you do that, you are breaking bonds. You break those covalent bonds that we talked about where the electrons are shared back and forth. When you break covalent bonds, you release energy. Energy gets released at the end. So we're going to talk about our energy molecule at the end, ATP. ATP is doing this all the time. It'll be ATP, then it breaks the bonds and it becomes ADP. It's a rechargeable battery force. It's going to be really cool. So we are doing catabolic and anabolic. Anabolic, small things, and we build them up to create a bigger thing. And energy has to come in to do that, to create the bonds. In catabolic, cats are going to take a big thing and break it into smaller things in catabolic. Catabolic, big things, smaller things. And when we break the bonds, we break those covalent bonds, it releases energy. And that's going to show up at the end. So both of these, catabolic, anabolic, they're happening all the time in your body. And both of these happening all the time in your body is metabolism. So let's talk about how we can create macromolecules. That's going to be anabolic, where we take small things and we make a bigger thing. Our first molecule we're going to talk about are carbs, carbohydrates. Carbs are your friend. Everybody needs carbs in part because your nervous system runs on carbs. Your brain and your nervous system loves carbs. That is their energy source, are carbs. So with our macromolecules, all of these guys are going to have a monomer, the simple building block, and we put them together and it will build a polymer. Mono means one, polymer means lots, many. So carbohydrates are going to be built out of carbon. All of our biological macromolecules will be built with a carbon backbone, a carbon base. When we learned about the valence shells on our Bohr model, we talked about two in the middle. And then as we get to carbon, carbon had four empty slots in its valence shell, which makes it kind of special that it's got these four empty slots there, which means it can bond four different places. An example of one of our most simplest guys out there is methane, where we have this one carbon molecule and it has the four hydrogens bound to those four empty slots. And now it's got a full valence shell here as methane. So, our carbohydrates are going to be all of these macromolecules are going to be organic molecules, meaning that they are carbon backbone, they have a carbon base, and they are going to have covalent bonds between the carbon and the other atoms involved in this molecule. Hydrogen is always one of them, though. You always have at least one hydrogen somewhere in an organic molecule. It's always got a carbon backbone, always, and it always has at least one hydrogen to make it an organic or biological molecule. So carbs are our first one. Again, they are the most abundant compound out there. Our carbs have come in a ratio. It's usually a 1 to 2 to 1 ratio with carbs. So our favorite one that I always like to go to is glucose, sugars that our body uses every day for energy. Glucose is C6H12, oxygen O6. And you can see that's the same kind of ratio. If for every one carbon, there's two hydrogens and there's one oxygen, a 1 to 1 ratio. These are going to have building blocks. All of these macromolecules will have building blocks to build them up. Um, with our building blocks for carbs, saccharide means sugar and mono means one. So these are one sugar molecules or monosaccharides. There are small little monomers or building blocks to make the polysaccharides. In the case of carbs, our monosaccharides, this example here is really cool because remember how we learned about isotopes, and those were the things that have different numbers of neutrons in their core, and isotopes had varying numbers of neutrons. Isomers, isomers are going to have that same chemical formula, but they are structured differently. So isotopes with the neutrons are different than isomers, isomers that have the same building blocks, but we take those building blocks and we build the structure differently with those. So in this case, these are monosaccharides that are also isomeric monosaccharides, meaning they all have C6H12O6. They all have 6 carbons, 12 hydrogens, and 6 oxygens in these molecules, but you can see they're built differently. Each one of these has a slightly different structure. Here's a hydrogen, here's hydroxide, here's hydrogen again. Here is hydrogen and hydroxide, so it's kind of similar in this fructose to glucose, but up above, we got a double bond here on this oxygen where we didn't have it before, where the double bond was over here with glucose. So every single one of these has the same number of carbons, hydrogens, and oxygens, but they're totally built differently. So that makes them isomers. And these particular isomers, glucose, galactose, and fructose, are all sugars. They're all simple sugars called monosaccharides. Um, More simple sugars. So we talked about these guys that were all isomers. There are more simple sugars out there. We are going to talk about DNA later, and DNA is made out of this simple sugar, deoxyribose. If we're talking about RNA, that we're going to talk about in the end of this video too, RNA is made out of ribose, the DNA and deoxyribose nucleic acid, DNA, ribonucleic acid, RNA, has ribose. So look at these two and let's check out the differences between DNA and RNA and the sugar backbone that's part of them. Ribose, hydroxide, hydroxide. Deoxyribose has deoxy in the name, which means without. It's pulling the oxygen out. So let's look at why it's called deoxyribose. Here's our hydroxide, just like in ribose. So that looks totally the same. But look at this. It's missing the oxygen over here. So the oxygen's missing, therefore, that's why it's called deoxyribose. And that is the sugar that you're going to find in DNA. And ribose is the sugar that you will find in RNA later when we talk about nucleic acids. Right now, we are just talking about the sugar part of the molecule, and these are the simple sugars that are called monosaccharides, and we will use them as building blocks for other structures. So if it's just one sugar, it's a monosaccharide. If we put two sugars together, we got two sugars. Disaccharide is two sugars. So if we have a glucose and a fructose, and we want to put these two little sugars together, we can totally do that and make a disaccharide. When we do that, they're going to bond together. We're going to take our two monosaccharides and we bond them together. The bond, if it's talking about two sugars, that bond is called a glyco. It's got sugar in the name. Glyco is sugar. Glycosidic bond bonds these two monosaccharides together to create a disaccharide. So here's our glycosidic bond down here. These are two sugar unit molecules. So we can make lots of different disaccharides. There's some common ones out there. So we have a glucose and a glucose. And if we have two glucose together as a disaccharide, that makes maltose. Maltose is malt sugar. Here we get glucose, glucose equals maltose. And that's our picture of it over here. If we have um, glucose and galactose, it's got lact in the name. Lact. What does that make you think of? If somebody is lactose intolerant, what can't they drink? Glucose and galactose is milk sugar. If you are lactose, if you are lactose intolerant, you don't have the enzyme lactase that breaks down lactose. And therefore, your gut bacteria breaks it down for you, and that results in an unpleasant experience. Versus if you had the enzyme lactase. If you have lactase, you can break down lactose, no problem, and it's fine. Ice cream and cow milk are your friends. If you don't, though, you may need to try a different type of milk or the kind of milk that has the lactose removed. Some milks I've seen lately in the stores have those, or you could try a milk alternative like almond milk or soy milk or any of those things out there. But this is lactose. It is a disaccharide. It is made out of glucose and galactose to make lactose. And if you have the enzyme lactase, you can break it down. If you don't have the enzyme lactase, um, you can still break it down, but you don't do it. The enzyme doesn't do it. The gut bacteria in your intestines do it. And the other disaccharide up here, the other two sugar molecule up here, fructose and glucose are fruit sugar, and our sugar, our animal sugar glucose is going to be table sugar right there. So that is sucrose. That's this guy down here. So one sugar, one simple sugar, monosaccharide. We pair them up with a glycosidic bond and we got two simple sugars together. That makes a disaccharide, two sugars. And let's keep going. What if we did this over and over and over again in a chain? Then we'd make a polysaccharide. So poly means many and many sugars. So we have three polysaccharides that we need to know about. On our polysaccharides, some of these guys are going to be sugar storage molecules. And I need you to know which one is a sugar storage molecule for us, for people, for animals, and which one is a sugar storage molecule for plants. So for plants, sugar storage molecule is starch. If you enjoy potatoes, French fries, that kind of stuff, potato is a pretty starchy plant. So it has its sugar storage in the form of starch. That's how they store their energy. This is plants storage molecule. We animals are going to store our sugar as glycogen. So the way it works, you're going to ingest your food. I just had dinner before I made this video. I ate my dinner. My body has broken it down, and it took all these complicated molecules in my food, and it's broken it down to the really simple itty bitty parts, right? Into these monomer, into these simple units. And I'm going to bring that into my bloodstream through my intestinal system. It's going to go into my bloodstream, and then I'm going to, convert it into whatever I need those, what I'm going to use those little monomers for. The sugars, I can take those simple sugars and I can squirrel them away. So if I don't need the glucose right now because I am literally sitting in my computer talking to y'all and I'm not out there exercising or anything right at this moment, it will take those sugars that I've eaten from dinner, store them away. It stores them away as polysaccharides, and that sugar storage molecule is glycogen. Glycogen is our sugar storage molecule, and it is, you can see, lots of monomers put together to make this polysaccharide. And so I'll store those sugars away in my skeletal muscles and I store them away in my liver, and then they're there for me if I need them. So I'll store them for about 18 hours. So that's how long your sugar storage is. You have short-term energy storage in your skeletal muscles and your liver for the food that you have been eating, the drinks that you've been drinking in can take your sugar storage molecules of glycogen and keep them for you. Because maybe, 5 hours from now, I am hungry. I've used up what I brought in and I'm hungry for more and my blood sugar gets low. If my blood sugar gets low, I'm going to look at this glycogen And I am going to use, instead of insulin that created this, insulin helps to create this polysaccharide and store the sugar away. If I'm hungry, I'm going to bust this stuff up. I'm going to use glucagon hormone to break that up. And then when I break up the polysaccharide, I'm going to have lots of little simple sugars floating around in my bloodstream, and it's going to raise my blood sugar again. When you break up in the morning and you're hungry, your blood sugar's low, that's what we're going to do. We're going to break apart our glycogen and raise our blood sugar. So glycogen is animals' polysaccharide sugar storage molecule. If we were plants, we would use starch as our sugar storage molecule. Another type of sugar storage molecule, another type of polysaccharide, I take that back. Another type of polysaccharide is cellulose. So most of us have heard of fiber. You know, you always hear about how fiber is good for you. Fiber in your diet is good for you, and it's true. Fiber in your diet is good for you. You cannot digest cellulose. We cannot eat, wood and be able to digest that. Cellulose is going to be found um, in the plant's cellular makeup, and we can't break that stuff down. So plants create this polysaccharide, but we can't break it down. We can break down starch, we can break down glycogen, we cannot break down cellulose. So that is fiber. So fiber is good for us in our diet. It's, we have soluble and insoluble fiber, but we can't break it down. Even termites can't break down cellulose, and termites are notorious for eating wood, right? But they have some bacteria and archaea that live in their gut, and that breaks down the cellulose. So even they need help to break down cellulose. So fiber is not our sugar storage molecule that we are going to break down. Fiber is plants, and that is separate from starch. That is going to be their energy source. Energy. Sugar is our short-term energy storage molecules. Short term. Again, like 18 hours. I can go into my sugar reserves, my skeletal muscles, break those down, and use that later, 3 hours from now when dinner wears off and I start to get hungry again. More pictures of polysaccharides. So here's a starch. Which one was it? Is it plant or is it animal? Sugar storage molecule. You need to know. Glycogen. Which one, plant or animal? It's us. Glycogen is us. And cellulose was that fiber that is found in plants, but it's not their sugar energy storage molecule. It is a polysaccharide, but it's not an energy source. And chitin is another type of polysaccharide. You will find chitin in fungi. Fungi have chitin, and bugs have chitin. Bugs have a skeleton, but it's not inside their body like ours is. It's on the outside, right? Anybody who stepped on a bug, like a roach or something, has heard it crunch. Oh, that is their exoskeleton, and that exoskeleton is made out of chitin, which is a polysaccharide. So polysaccharides are long molecules of sugars, and those long molecules are just made out of individual units, those monomers. And in this case, if we're talking about sugars, they're monosaccharides. Benefits of carbs. Again, carbs are not the devil. My parents used to subscribe to a diet called Atkins back in the day. Today it's kind of reformatted and it's called keto. And it's a very low carb diet. And there are benefits to restricting your carbs slightly. I mean, you don't want to go overboard on too many sugars, especially simple sugars. That is not healthy for you. But whole grains are good for you. If you're taking a whole grain bread, if you're taking an oatmeal, those whole grains are better for you in your digestion. They take longer to digest. If I was going to have a choice of Fruit Loops or a bowl of whole grain, a bowl of granola or something like that with nuts and carbs and the oats and stuff in there, that takes a much longer time to digest than Fruit Loops or Captain Crunch or whatever my kids like to eat. Those are processed carbs, processed sugar, and they will spike your bloodstream. So your blood sugar goes woo like this, and then you crash. If you have complex carbs, multigrain bread for your toast, whole grain cereal, that kind of stuff, the breakdown of the carbs is much slower because it takes a longer time to process all those carbs, and it doesn't crash down like before. So you can have a nice steady burn on those. So carbs are different. You want your complex carbs, not your simple sugars. Complex carbs are what you want. You want whole grains because they are going to have fiber that we saw when we saw the cellulose. So fiber is going to lower your sugar consumption. It's going to keep you regular. It helps you go to the bathroom. Sounds gross, but it's essential. You need to be able to do, you know, you need to be able to go to the bathroom. So fiber helps with that. It's also going to lower cholesterol because as the fiber is moving through your body, it's going to help remove cholesterol that builds up in your bloodstream that can cause plaque and heart disease later in life. You don't want that stuff. So, fiber helps lower cholesterol. It can also prevent certain forms of cancer, like colon cancer. So, it's cleaning the blood out of the cholesterol out of your blood. It is um, going through your intestine and helping with your digestion. It's keeping you regular. It's helping prevent certain types of cancer like colon cancer. Um, Multigrain, complex carbs, they are your friends. They are your friends. So carbs are not the devil. There are good carbs out there. So again, whole grains, fruits and veggies, those are all great. And lean meats are a source of protein that we're going to talk about next. Okay, so those are carbs. Let's move on to the next macromolecule, which is lipids, your fats. Lipids. So these guys are going to be non-polar. Remember, we talked about water was polar and water was super awesome for all of the reasons that water is awesome for. And if you liked water, you were hydrophilic. If you did not, you were hydrophobic. You don't like water. So you're not going to bond with water. What do lipids do? We said sugar was short-term energy sources. Lipids are going to be long-term energy storage. So if I ate, a whole pack of Oreos one day and all those sugars got stored up, ready for me to use, but I did not use up all of those sugars. Eventually, I'm going to convert those sugars into lipids. They are going to convert into fat, which will be our long-term energy storage. Fat is a great insulator to help protect you from the environment. If you ever hear about bears, we have bears, we have black bears here in Georgia. They are going to hibernate every winter, right? And what does a bear do before it hibernates? It packs on the pounds, it packs on the fat because that helps insulate them. It is a great building block for hormones, different hormones. Some hormones are made from proteins and some of them are made from lipids. Hormones are going to be really important for all sorts of communication throughout your body. Hormones for your fight or flight, autonomic nervous system, hormones like sex hormones for testosterone, estrogen, all sorts of important hormones are built from lipids and proteins. It is also a component of the plasma membrane, and plasma membranes, every cell has a plasma membrane. You've got to have lipids. You need to have phospholipids to build your cell membranes. So they're important. This macromolecule is very, very important. We need lipids too. They come in a different variety. They're not just like simple old monomers out there. They come in different structures. So we have fatty acid chains, we have glycerides, we have phospholipids, like we talked about with the cell membrane, and there are steroids and eicosanoids too. We have this adorable little otter, and this guy lives in the water. But what's cool about the otter is he's going to have glands that are going to release oil that helps protect his coat, that helps protect him to keep the water off of his skin underneath the fur. So he sheds water and he can stay warm and insulated because he coats himself in this oil. And we know oil is hydrophobic, it doesn't like water, and it's going to keep him protected and insulated. It's like his little raincoat. All right, so lipids, they are going to have different structures. With our fatty acids come in chains. And here we have a saturated fatty acid and we have an unsaturated fatty acid. I'm going to want you to know the difference between saturated and unsaturated fatty acids. So if we look at our saturated fatty acid over here, hopefully we can see that this guy is in a straight line, right? He's going relatively in a straight line here. Carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. Hydrogens are everywhere and hydrogens are literally saturating the carbon. Since it is in a straight line, these guys can build and become pretty thick molecules. And these thick saturated fatty acid chains are going to be, when we talk about fats and fats that are at room temperature, those are saturated fatty acids. So think about your lard, think about your bacon grease. Those thick, saturated fatty acids are solid at room temp because they have these long structures that stack nice and neat. Saturated fatty acids are solid at room temp and they last um, longer than unsaturated fatty acids. So our unsaturated fatty acids, it kind of looks the same. This chain sort of looks the same, but it is different. And there's a key difference. It's straight, it's straight, it's straight, it's straight. It's saturated with all these hydrogens. But at this point right here, this is the most important thing that's happening on this fatty acid chain is that it has a double bond. This double bond forces this molecule to change shape. It changes shape. So since we're missing a hydrogen over here, we don't have carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, and this pretty little row of carbons and hydrogens, we missed a hydrogen. And since we missed a hydrogen, those carbons are going to double bond, and that double bond bends this molecule. So it is bent. It is not straight anymore. This is a bent molecule because of that double bond right there. And this is an unsaturated fatty acid. Solid unsaturated fatty acids, because they are bent, they don't lay flat. They are not flat, they are not solid at room temp. These guys are liquid at room temp. So usually examples of these are your oils. These are your oils like your olive oil, grapeseed oil, whatever oil you like to cook with at home um, are unsaturated fatty acids. Again, the molecules don't lay flat. They are bent because of that double bond. They are missing hydrogens, and therefore they double bond. Instead, the carbons double bond when they miss the hydrogens. That double bond changes the shape of the molecule. It doesn't lay flat. These guys don't buddy up next to each other, and they are liquids instead of solids. Liquid at room temp are unsaturated fatty acids. They are oils. Solid at room temp are saturated fatty acids. Examples of those is bacon grease, lard, coconut oil, all of that stuff is solid at room temp. So know the difference between saturated and unsaturated fatty acids. Here is a triglyceride. I was telling y'all about cholesterol and fat and stuff that can build up. So this is a triglyceride. It has fatty acid chain, and here is 3 saturated fatty acid chains dangling off of this bad boy. And this part here is a glycerol head, so that's why it's called a glyceride. This has 1, 2, 3 fatty acid chains coming off of it, so it is a 3. It is a triglyceride. When you bring in triglycerides in your diet, you can break them apart and they will lose a fatty acid chain and it's a diglyceride. Lose another fatty acid chain and it's a monoglyceride. So this is an example of a triglyceride. I told y'all that phospholipids are important for cellular membranes. We're going to get to that later in another chapter. Cellular membranes are built out of 2 layers of phospholipids. So here's our fatty acid chains, here's our glycerol head, but there's also, this is the all important part to why it's called a phospholipid, is the phosphate group right here. This is the phosphate group that makes this a phospholipid. And then another type of fat out there are going to be our steroids. That's this guy, and they usually have a ring structure. Your steroids are going to have a ring-like structure. Steroids, they are going to have this basic ring structure. Your waxes are going to have a hydrocarbon chain like this. Some of your examples of waxes, beeswax is the first thing that pops into my mind about waxes. Plants produce wax too. If you've ever seen leaves that look kind of shiny, some leaves will produce a waxy coating in there to help with evaporation so they don't lose all their water loss that way. Those are lipids. Waxes are another type of lipid. If you want to check out more about lipids, check out Wisconsin's page about it, and you can learn more about saturated and unsaturated fatty acids. Trans fats. Trans fats has been talked about quite a lot in the last decade or so. We've been more aware and conscious about trans fats. What are trans fats? So trans fat is when you are going to take the molecule, and here, remember we talked about how if the Saturated fatty acid lost its hydrogens, it missed them, and it formed a double bond, and that changed the structure and it made it bent. Well, here we have our bent molecule, and normally you would say that this is an unsaturated fatty acid and it's going to be, a liquid at room temp. But if we change the hydrogen to the other side, cis is same, cis means the same, trans means the opposite. Usually we think about trans, you hear it most often when we talk about gender. Um, if somebody is transgender, but we can talk about it in chemistry too. And it just means that it's on the opposite side of the chemical bond on this double bond. So here is our hydrogen, and the other one was over here, and it moved to the other side, to the opposite side, and this is a trans fat. So when we make it on the other side, look at it. Now it lays flat, and now we've changed something that would have been a liquid at room temperature to a solid at room temperature. This is going to last a lot longer on the shelf. So a lot of trans fats, these guys are man-made when we manipulate the lipid, and it's usually done to extend the shelf life of a product, of some sort of food product, is where you see the trans fats. And so now they're labeled, and you can see when that has been altered in your food and decide whether or not you want to take that into your diet or not. But this is what it is. It's when they change the structure of the lipid. Either they coat it in hydrogens, or they change the structure and manipulate it so that it is solid at room temp and lasts longer. All right. Proteins are going to be another macromolecule. There are 4 total macromolecules we're going through. This one's number 3. Proteins. Proteins are important too. All these macromolecules are important. You're going to need to know what the macromolecules are, what their job is, descriptions about them, why they're important, all that good stuff. So with proteins, we use these all the time. Proteins can be built in and form enzymes. An example of an enzyme that we just talked about is lactase. Lactase breaks down lactose. Enzymes are great. They are going to help you perform a reaction with less energy needed. We're going to go into those in a little bit. They can help with transport. Hemoglobin is going to transport oxygen, and that is a molecule inside your red blood cells, and it is absolutely important. Other things, proteins are important for structure. Actin is a fiber inside your muscles. Every time your skeletal muscles move, your heart muscles move, you have actin and myosin fibers in there, even your smooth muscle, that allow your body to do all the things it does. Hormones, we talked about. Proteins are helpful with defense, your antibodies, those antibodies against all of the different diseases out there that your body has, your immune system. Those are proteins. You need these guys. Contractile proteins, structure proteins. Um, Proteins are essential. They are helpful. What is a protein? Well, we talked about those simple building blocks for sugars. Those were monomers, and those monomers were monosaccharides if we're talking about sugars. If we're talking about proteins, the the monomer for a protein monomer is going to be an amino acid. Amino acid. Every amino acid is going to have an amino group over here and a carboxyl over here. So amino acid. Every amino acid, and there are 20 in your body. Every amino acid is going to have the same head. It's going to have the same part right here, all of them. The tail differs. So there are 20 different ones, and they're all going to have a different tail. So this one's tail. Here's valine's tail over here. Lysine looks like this. Every protein in your body is made with 20 different amino acids, and they're just put together in different ways. But these are the building blocks. Your building blocks for proteins are amino acids. Check out more about them there. Um, so we are going to uh, briefly chat about enzymes. We said that enzymes are necessary because they help you do things with less energy. You can break down lactose with lactase, and it's simpler to do with enzymes. We can see here's activation energy in purple, the energy needed to perform a reaction, to break sugar down, to get its byproducts, needs activation energy. And if we have an enzyme, we need less of the activation energy. It helps, basically. Enzymes help. I'm not going to go into all the details here in the slide, but know that they help you get to your product to perform your reaction with less in energy. Here is an example of, here's our substrate, hitting up the active site, which is this place right here on the enzyme, and it's going to end up producing, here's our products at the very end of this reaction with our enzyme. So if we were breaking down peroxide, um, this could be our, our water molecule and our oxygen gas bubbling off there when we took in peroxide on this end. Enzymes help us perform a reaction with less energy in general. Protein structure. We definitely need to talk about protein structure and how we build them. So later on, we're going to talk about central dogma and how a cell performs its duties. And a cell is going to have DNA on the inside, and it's going to use that DNA to code a message called RNA. And based on that message, it's going to build proteins. Proteins are made out of amino acids. So we have 4 different levels in protein structure. We have our primary protein structure when it is just simple amino acids strung together like beads on a necklace. That's the most simple we can get, is this amino acid primary structure. Then we are going to have the hydrogen bonds. When we talk about hydrogen bonds, where we have these weak electrical charges, a weak positive and a weak negative, and they're going to have this hydrogen bond going across there. Um, that is going to cause a certain shape, and it can be either be a helix, the alpha helix, which looks like kind of like a DNA molecule, an alpha helix, or it could be a beta pleated sheet. If you have a question on the test and it's describing a protein structure and that protein has a helical shape or a sheet, we're talking about the secondary protein structure, and that is caused, the helix and the sheet is caused by the hydrogen bonds on those amino acids that are now interacting with each other. Now we're going to get even more complicated, and we're going to go to the third level, tertiary protein structure. And this is going to happen. We had hydrogen bonding in secondary. No, go back. These were the hydrogen bonds. And then all of those other tails are going to interact too. No matter what the remainders were, they interact. So I'm just going to write down tails are reacting. All of these chain interactions are forming. So this is where where you started so simple up here. This was simple beads on a string, amino acids strung together with their peptide bonds. When two monomers, when this amino acid is bound with this amino acid, it has a peptide bond. So in this guy right there, that is a peptide bond. And it's very simple at this level. And then it gets a little more complicated with the hydrogen bonds. And then this is as most complex as a single protein chain can be, is tertiary. So we have one chain, and it is not only starting to change shape with its hydrogen bonds, but all the tails, all those remainders are interacting too. We are in tertiary level, and it's real complicated, and it has a very distinct shape, and that's how that protein should remain in that shape. Shape is key. Shape is fundamental in protein structure. It's absolutely critical. When we have multiple chains interacting amongst each other, it is quaternary. Quaternary protein structure are going to be more than one amino acid chain. So an example of quaternary structure is hemoglobin. Hemoglobin has 4 chains, and it is a molecule found in your blood, in your red blood cells, and you have like 250,000 molecules of hemoglobin inside your one red blood cell. So you got lots of hemoglobin in there. And it has 4 chains for every hemoglobin molecule, and that is quaternary. So this is still a single chain in primary. Secondary is still a single chain, but it's in a shape of a helix or a sheet. Tertiary is still a single chain, but now it's got sheets and pleats and all sorts of strange structures. And then quaternary, multiple chains for quaternary structure. Again, with proteins, shape is important. You have to have your protein shape because that's what helps the protein do what it does. If the protein changes shape, it shouldn't. But there are things that can cause proteins to denature when a protein changes shape. So I like to think about it as when you're cooking eggs, if you eat eggs for breakfast in the morning, you're scrambling eggs, or if you've at least, watched the video about it, crack the egg, put it into the hot plate, and temperature is then going to change that liquid proteins, the albumins in the egg, and then they are going to change and they are going to be solid, and you can never go back. You can never change that protein back to its liquid form. No matter what you do to it, it's still changed. It's fundamentally changed. It has denatured. It has changed the protein structure. So different things can cause this, but temperature is one of those. It's another reason why people can suffer permanent damage if their temperature gets too high. If your body temp gets too high, you might suffer permanent damage. Those proteins in your body may become denatured as a result of that. So we don't want proteins to change shape. Their shape is very important. And then we mentioned hormones. Hormones are important. They were signaling molecules, sex hormones, your fight or flight hormones, hormones to help you bring in more water and maintain your hydration. There's all sorts of hormones in your body, and they are built off of proteins and lipids. The last macro that we need to talk about in this video is nucleic acids, the last one. Nucleic acids are important. They have a monomer too. Monosaccharides for sugars, amino acids for proteins, and nucleic acids are nucleotides. Nucleotides have three parts. There is a sugar, and we said it could be deoxyribose or ribose. It's going to be some kind of sugar. And then it's going to have a phosphate. So sugar, phosphate, and a nitrogenous base of some kind. There are going to be four different bases if we're talking about DNA, and four bases if we're talking about RNA, purines and pyrimidines. But this is your monomer, your single unit for nucleic acids, and it is a nucleotide. Nucleotide. What are the nitrogenous bases? Well, here's your choices. We always have adenine and guanine, A or G, and those are purines, and they will always bond with their pyrimidine best friend. That's how they bond. So they will bond with across the way. They will bond. They're going to have hydrogen bonds, and they're going to zip the DNA molecule together. So if we have an adenine, it always binds, if we're talking about DNA, with thymine. A's always bond with T's. Adenines are always with thymines. Guanine is always going to bind with cytosine. G's are always with C's. And this is DNA. That's how they roll. T's and A's and T's, C's and G's with C's. Those are always going together. They are the best friends. They are going to bond across the way. So here is our sugary nucleotide. We've got our phosphate. Here's our sugar, our pentose 5 carbon sugar, and our nitrogenous base. And here is one nucleotide, and it's going to have another nucleotide and another nucleotide with our phosphodiester bonds. DNA structure is a double helix. A double helix, it's going to have those complementary base pairs between the A's and the T's and the C's and G's. We need to know who bonds with who. You will have questions about that. You may have an example. Say I'm just going to make stuff up. Maybe I have DNA and it is, sure, I have a DNA strand and this is one side of the DNA, and I need to make the other complementary side of DNA. Well, who matches with who? If I remember the word at, A's and T's always go together, and that means C's and G's are then going to go together. So I have a C here. It's going to bond with its best friend, G. G is going to bond with its best friend, C. T's best friend is an A. C, again, we've got another C, so it goes with G. And A's best friend is a T. That's DNA to DNA. What if I was going through central dogma and I went from DNA to RNA? I needed to make messenger RNA. What if I was making messenger RNA? Like in the COVID vaccines right now, those COVID vaccines are using messenger RNA in the vaccine. That's what they're doing. So if we had C, G, D, T, C, A, DNA, and we wanted to make RNA, there's one key difference. There's one key difference. It's really the same thing, except for RNA does not use T, does not use thymine. It's going to use uracil. Uracil is used in RNA. So RNA is going to use uracil. So let's write it out now. Now we know that we're not going to use T's, we're going to use U's. We can do this. It's the same thing. C, best friend G. G's best friend C. T, best friend is A. C's best friend G. And A, where normally I would put a T, but if we're making RNA, we're going to swap out the T and we use U. It's as simple as that. Now I have made us a complementary strand of RNA. So on the test, you will need to know who bonds with who. Can you make a complementary strand given an example strand? We need to know who buddies up with who. DNA structure is going to have all of these nucleotides stacked on each other. They're going to have the sugary backbone, the sugary phosphate backbone. It's going to run anti-parallel, or one side is going to be 5' and 3'. We'll go over more of this later. The other side is going to be 3' to 5'. So that's why they're opposites. They're called anti-parallel. We'll go more into this later. I'm not going to get into too much into DNA, but I do want you to know that it is double-stranded. You can see that there are two strands here. They have little hydrogen bonds. All of this are hydrogen bonds here going between the nitrogenous best friends, between the C's and the G's and the A's and the T's. RNA, instead of being a double strand, it is a single strand. Here's our messenger RNA. This is central dogma, DNA, RNA, and amino acids right here. These are all little bitty amino acids buddied up together with peptide bonds. To make RNA, instead of deoxyribose as that sugar, you're going to use ribose. It's simple. You're going to have a phosphate. You're going to have your 5-carbon sugar, and in this case, it's ribose. And then you are going to have some type of nitrogenous base. C, G, A, or U over there. But it will be in a single line, a single strand, instead of a double helix. If we want to compare them, again, the sugars are different, DNA versus RNA, deoxyribose versus ribose. Bases are different, thymine on DNA, uracil on RNA. DNA is much, much bigger. Your DNA inside your cells, that is you. It's going to have your coding, your genes for coding stuff. It's going to have non-coding stuff in there. It is long, long, long. So more than 45 million. RNA are messages. They are just short and sweet messages to build out the proteins that your body, your cell needs to do things. So they're not near as long, 100 to 50,000 nucleotides in each one. It is a double helix. DNA has a double helix on there. Oh, that's terrible. That's totally terrible. It's terrible. While RNA is going to have a single strand. DNA is your genome. It is all of your genetic information in your 26 different, 23 pairs, 46 different chromosomes that you have. Your entire genome is in there. RNA is going to help. The end result is protein synthesis. It's going to be DNA to RNA to amino acids, protein synthesis. That's its goal for RNA. It's just a message to create proteins. The last nucleic acid we're going to talk about is ATP. That's our energy molecule. ATP is going to be adenosine triphosphate. Adenosine triphosphate. It has a sugar, ribose, and it's going to use adenine nitrogenous base. And instead of one phosphate like our other nucleotide, it has three phosphates, hence the triphosphate name. And ATP is our energy currency molecule for sugar, not for sugar, for energy. We are going to use ATP, and it will go from ATP with three phosphates. And then when we need to break off the third phosphate, what we said that when we break covalent bonds, it releases energy. And so we will go from ATP triphosphate down to a DP, which is a diphosphate. We've broken off that third phosphate, and we can harvest that energy to perform whatever metabolic processes we need to do. So ATP is another type of nucleic acid that our body uses, and it is our energy currency. Later, when we get to cellular respiration, we will be talking a ton about ATP. All right, so this was a really long lecture about macromolecules. It's a lot to bring in. We are trying to stay on the surface of this information. Let me know if you have questions. I realize chapter 2 is a lot to absorb. So please let me know if you have questions on this stuff.